Welcome to our video that reviews the process of translation, a key step after transcription in a cell's grand effort of creating a protein. Now let's talk about stage 2 of protein synthesis, which is translation. Ribosomes. Let's just look a little closer at the structure of ribosomes. These are the sites of protein synthesis, which is why it's really important we talk about them today. When we're talking about translation, we're going to be synthesizing these proteins now. So ribosomes have two different sizes. There's the large subunit, which is called 60S, and the small subunit, which is called 40S. That S actually refers to the sediment position in the centrifuge tube. So when these ribosomes are put in a centrifuge tube and they spin around and divide its constituents based on its density, the large ones end up in a position at the 60 in the tube and the smaller ones at 40. So these combine together to form an intact ribosome. When the intact ribosome all comes together with the large subunit and the small subunit, they call it an 80S. This also combines the rRNA and protein all together, which forms that intact ribosome, the 80S. Now, the reading frame and mRNA, when we're talking about mRNA, and this is what we're going to be reading to translate it into the specific amino acid sequence that makes the polypeptide chains, the reading frame is very important. Okay, so sometimes we'll get a reading frame which just shows a bunch of bases, as you can see at the top line there. Okay, if you space them out a little after every 3 and you make distinct codons, that helps in properly reading it. So that's a good tip to use. Oftentimes in textbooks, we might see them already in triplets to be read as codons. Now, it's really important to note that if you accidentally miss one of the bases when you're reading it, or you miss an entire codon, that ends up shifting the reading frame over. And that could end up making new codons, which makes new proteins. And then the previous function of the protein that was supposed to be made doesn't end up being formed and actually working properly. So here's a very simplified example or overview of translation. We have that large subunit and small subunit that come together to make a ribosome, and it moves along the mRNA from the 5' along to the 3'. And what we see as an output is the polypeptide chain that forms of amino acids bonded together. So eventually the ribosome will reach the termination codons and translation will stop, very similar to transcription. Okay, but this is actually happening on the mRNA strand this time and making amino acids as the outcome. And then that polypeptide can now be further processed in the cytoplasm to be folded and interact with other polypeptide chains to finally make that eventual quaternary structure of a functional protein. So we've mentioned tRNA. Let's take a closer look at the details of tRNA here. So tRNA is a small single-stranded nucleic acid. It actually forms a clover leaf in shape, which delivers the correct amino acid to the ribosome. The anticodon end, where you can see at the very bottom there in the darker green, has 3 bases, as you can see. So on this example, it says AUA. And it recognizes the codons of the mRNA. Okay, it's called the anticodon because it has complementary bases involved in the anticodon of this tRNA. The opposite end carries the specific amino acid. Okay, so you can see at the very end there, it says it's the acceptor site. So right up at 12 o'clock there, the acceptor site of an amino acid. In this case, it's tyrosine because the anticodon codes for that, takes it from the coding section of the mRNA. Recognition of anticodon to codon via base pairing occurs. It's very similar to complementary base pairs with DNA and RNA. In this case, it's RNA with RNA, right? We have the mRNA. It's going to pair with the tRNA with the anticodon. Now, 20 to 61 different types of pRNA exist, depending on the organism, to code for the 61 possible codon combinations for the amino acid. Not 64, since remember there are 3 stop codons. So those are release factors. They don't actually code for specific amino acids. Now there's aminoacetyl tRNA, which is a tRNA molecule which contains a specific amino acid. And we say that this is charged. There's also aminoacetyl tRNA synthetase, and this is the enzyme responsible for attaching the appropriate amino acid to its corresponding tRNA, which we saw happens up at the top there. So there are at least 20 different enzymes which are specific for a particular amino acid and in turn, a particular tRNA. So let's look at the details here. You can see in A that the first codon is recognized by the ribosome there. Okay, so we have the codon AUG in this first example. You can see that that's the first in the sequence, the first codon in that sequence there. That will be translated. The AUG ensures correct reading frame as every protein will begin with the MET. Remember, we talked about the start codon, which codes for MET, M-E-T. And you can see there in the ribosome, we have that tRNA. It's shown in a blue glob of sorts, okay? It's labeled as tRNA. You could see at the bottom there, it's the anticodon, and it's showing UAC, okay, which is the complementary pairs for AUG, which codes for MET. And you can see heading off the ends of the tRNA there, opposite of the anticodon end, is that amino acids that tRNA specifically carries. So you can see the MET there. This tRNA enters the P site where the anticodon comes into play with the mRNA strand. Okay, and that P site is called the peptide site. And we know that it reads it from a 5' to 3' direction. So you can see how it's going to start at the AUG, and it's going to bring in that net into the P site. And then it's going to bring in another tRNA with the complementary anticodon for GCA, which will bring in that appropriate amino acid and so on, moving rightwards in this example. So B, we can see here comes the second and the third amino acid based on the codons in the mRNA sequence. So the second tRNA carries a specific amino acid and enters the second site in this ribosomal complex, which is called the A site, which is an acceptor site, and it's bringing in alanine. You can see there ALA. Okay, so there's another tRNA coming in, bringing in another amino acid. And then we see that MET and alanine are bound with peptide linkages. Okay, peptide bonds form between amino acids. We know this. And then the amino acyl group will be broken as well, okay, to be able to break it off from the tRNA. Then as we keep going, the ribosome shifts over to the next codon. Okay, so it's shifted. We now have the peptide bond between MET and ALA, and we're coming in with isoleucine, ILE. The uncharged tRNA leaves the P site in this case. The tRNA in A site moves, it shifts into the P site as it's moving across there. And a new tRNA with another amino acid moves into the A site. Okay, so the amino acids the tRNA is shifting always into the P site as a new one comes into the A site as the ribosome keeps moving along the mRNA from the 5' to 3' direction. The amino acid is determined by the anticodon-codon complementary pairing, again, as shown before. So now we're at AUA, which the empty codon for that is UAU, which is found on the tRNA, which brings in isoleucine there. And what do you think is going to happen between the amino acids in the P site and the A site, the alanine and the isoleucine? Well, if you look over at D there on the right, they are going to form another peptide bond. And the ribosome shifts, another tRNA can come into the A site, and this is bringing arginine, and this happens over and over and over again. Okay, and this is how the polypeptide chain will grow with multiple amino acids being bonded by peptide bonds there. This process continues until the ribosome reaches the termination code, that stop codon. And then released factor proteins will aid in the dismantling of the ribosome mRNA complex, releasing that final long polypeptide chain. So uncharged tRNA molecules will go back into the cytoplasm to pick up the correct amino acid for further synthesis of proteins. The three stop codons, as mentioned before when we looked at that table, it's UGA, UAG, and UAA. It's important to know the stop codons, as well as the start codon, the universal start codon. Further processing as the primary sequence protein may be necessary depending on its function. Sugars or phosphates can be added called glycosylation and phosphorylation, and or enzymes can cut the peptide at specific sites, or some of the amino acids in the chain may be altered by Golgi apparatus. In eukaryotes, the first amino acid to enter the P site will always be met, methionine. In prokaryotes, methionine is tagged with a formal group. The first amino acid to enter the prokaryote ribosomes is formal methionine because it's tagged. So they call that F-met